Okay, this is Chapter 5, the structure and function of large biological molecules. I'll try to get through this a little bit more quickly. It's a little bit of a longer PowerPoint presentation, but I'll try to make it as brief as possible so that you can get through this in 15 or 20 minutes if you'd like to watch it. Okay, here we go. So the molecules of life. All living things are made up of four classes of large biological molecules. They are carbohydrates, lipids, proteins, and nucleic acids. Macromolecules are large molecules, and they're more complex, and large biological molecules have unique properties that arise from the orderly arrangement of their atoms. Okay, a polymer is a long molecule that has many similar building blocks. Those repeating units that make up the building blocks are called monomers. Carbohydrates, proteins, and nucleic acids are polymers. Enzymes are specialized macromolecules that speed up chemical reactions such as those that make or break down polymers. A dehydration reaction occurs when two monomers bond together through the loss of a water molecule. Polymers are disassembled to monomers by hydrolysis, a reaction that is essentially the reverse of the dehydration reaction. If you look here at the top in A, you can see that the hydrogen from the short polymer in the hydroxyl unit for the unlinked monomer will form a water molecule and be removed, and thus allowing two opportunities on the short polymer and the unlinked monomer to bond to each other, forming a longer polymer. That's a dehydration reaction. The other thing, the opposite thing, is called a hydrolysis reaction. Hydro means water, of course, and lysis means to break down. So by adding water back in, we can give them the H and the OH units back, and therefore it can break the polymer down into the individual polymers, smaller polymers, or monomers as it may be, and by giving them their hydrogen and hydroxyl units back as appropriate and forming two independent molecules. Now that's called a hydrolysis reaction. Okay. Had to get through that slide here. Here we go. Okay, so a cell has thousands of different macromolecules. Macromolecules vary among cells of an organism. They vary more within a species and vary even more between different species. A huge variety of polymers can be built from a small set of monomers. Carbohydrates include sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides or simple sugars, and carbohydrate macromolecules are polysaccharides, which are polymers composed of many sugar building blocks. This is the first of the four types of major classes of large biological molecules, carbohydrates. Okay, sugars are monosaccharides. They have a molecular formula that's usually a multiple of CH2O, so that 1 to 2 to 1 ratio between the carbon, hydrogen, and oxygen. Glucose, for instance, since it's got 6 carbons, it's going to have 12 hydrogens and 6 oxygens. It's the most common monosaccharide. And monosaccharides are classified by where the carbonyl group is located, whether it's an aldose or ketose, as you remember, whether it's inside or at the end of the carbon chain, or whether the number of carbons in the carbon skeleton makes a difference or not. More obviously, the more carbons there are, it's going to lead it to cause it to have a different change and different function. So here's a couple different diagrams you can see on the left side here. You have a triose. Tri means 3. Ose, ending in ose, means it's a sugar. So you've got 3 carbons. So triose, it's a 3-carbon sugar. Now, two things could happen. You have the double bonded oxygen at the end, or you could have the double bonded oxygen in the middle. The double bonded oxygens at the end is called an aldose or an aldehyde. If it's double bonded in the middle, it's called a ketose or a ketone. Okay, and you've got some other examples listed below. Though often drawn as linear skeletons, if you take one of these carbon chains and throw it into an aqueous solution, many sugars will form rings. Monosaccharides serve as a major fuel for cells and as raw materials for building molecules. So here's an example. You've got on the left side a 6-carbon, so that's a hexose. You've got the double bonded oxygen on the end. That means it's going to be an aldose or an aldehyde. And if you were to throw it in water, it'll start to wrap around itself, as you see in the center of the top middle right here. I'm pointing at it right now. That's starting to form a ring, and then you'll have some reconfiguration on this end here as it forms a ring. So this is a linear and ring formula of the same type of molecule. A disaccharide, di means 2, again, so you have a disaccharide when a dehydration reaction joins 2 monosaccharides. The covalent bond is called a glycosidic linkage, what actually causes this to happen. So here, I have the dehydration reaction occurring between this hydroxyl unit and this hydrogen, forming H2O, so that gets removed. This allows the opportunity between this open-ended electron here and the free electrons on the oxygen to actually bond. Okay, and that is going to be called a glycosidic linkage. And because it's between the number 1 and the number 4 carbon, it's called a 1-to-4 glycosidic linkage. And so in this diagram here, if you dehydrate glucose with another glucose molecule, dehydrate glucose with another glucose molecule, you'll make maltose. And here, if you dehydrate glucose with a fructose molecule, you'll make a new sugar called sucrose, which is common table sugar. And in this case, sucrose has a 1-to-2 glycosidic linkage. Polysaccharides are the polymers of sugars, have storage and structural roles. The architecture and function of a polysaccharide are determined by its sugar monomers and the positions of where the glycosidic linkages are. Now, for instance, starch is a storage polysaccharide of plants. It has glucose monomers. The plant stores surplus starch as granules within chloroplasts and other plastids. And the simplest form of starch is called amylose. As you can see here in the top right, you have amylose, which is unbranched. So these glucose molecules will be broken off sequentially, one after the next, so you get a slow release of energy. In amylopectin, you have some branches, you may have some minor branching, so you can actually break glucose molecules off one chain and the other chain at the same time, one by one. So it releases a little bit more energy faster than amylose does. Now, when you get to glycogen, you'll see you got heavy branching. So this is animal sugar or sugar storage in animals, and glycogens extensively branch, which means you can release energy from here and here and here and here and over down here, all at the same time. So by breaking them down one at a time, you can release large bits of energy in a very short period of time. Generally, it's stored in the liver and the muscle, and this can release rapid amounts of energy for us. And of course, since we're much more active than plants are, we need a bit more energy than they do. And finally, the third type is cellulose, which is actually forms into a microfibril. They have a different type of glycosidic linkage. They actually have a 1,4 glycosidic linkage, but it's in an alpha and beta format. And because of that, you're allowed to have horizontal and vertical bonding. And this gives cellulose its unique characteristics, the crunchiness that it tends to get, since it does form a matrix when it bonds. Okay, glycogen, as we mentioned a moment ago, is the storage polysaccharide in animals. It's stored mainly in the liver and muscle. Breaking it down by adding water in or hydrolysis of glycogen in these cells releases glucose when the demand for sugar increases. And the last one we looked at was the polysaccharide cellulose, which is a major component of the tough wall of plant cells. Like starch, cellulose is a polymer of glucose, but the glycosidic linkage differs. It uses that alpha and beta configuration. Okay, so here we go. You can see alpha and beta glucose. Here you got the two hydroxyl units on the same side, and alpha glucose and beta glucose. They alternate, and because of this, if you have a linkage here, this is like an alpha linkage, 1-to-4 glycosidic linkage, right? You have a straight chain here. This could be like glycogen or something like that, one of the branches, or amylose or amylopectin, where they're all sequential and all the hydroxyl units are on the same side. However, with starch, you have 1-to-4 glycosidic linkage of the alpha glucose monomers, which means that the hydroxyl units alternate between the alpha and the beta units, and this cellulose here, this causes the opposing hydroxyl units, causes it to have the ability to form vertical bonds as well as these horizontal bonds, giving it that rigidity. So, any rate, you've got some different combinations you can get between these. And make sure you take a look closely at that in the textbook. Okay, starch alpha configuration is largely helical. Cellulose molecules, the beta configuration, are straight and unbranched. Some hydroxyl groups on the monomers of cellulose can hydrogen bond with hydroxyls of parallel cellulose molecules. Now, enzymes that digest starch by hydrolyzing alpha linkages can't hydrolyze beta linkages in cellulose. The cellulose in human food passes through the digestive tract and is known as insoluble fiber. Some microbes use enzymes to digest the cellulose, and many herbivores, like cows and termites that eat grasses and hays, have a symbiotic relationship with these microbes, and thus they're able to break down the grasses that they eat and derive energy from them, whereas we cannot because we do not have those microbes. Chitin, another structural polysaccharide, is found on the exoskeleton of arthropods. Chitins also provide structural support for the cell walls of many fungi. Here you can see, if you're familiar with the cicadas or cicadas, I don't know how they pronounce it here, but you see those brown shells left on your fences and houses. When insects and such or arthropods emerge from them, those hard shells that are left behind are generally made of chitin. Okay, lipids, the next class. So we went through the carbohydrates, which were starches, glycogen, and cellulose. Now we're going to take a look at lipids, are one class of large biological molecules that do not include true polymers. The unifying feature of lipids is that they mix poorly, if at all, with water. Lipids consist mostly of hydrocarbon regions, and the most biologically important lipids are fats, phospholipids, and steroids. So again, three classes in this, three subgroups in lipids. The first subgroup, fats, are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. The fatty acid consists of a carboxyl group attached to the long carbon skeleton. So here, if you look at the top, you can see this glycerol head here. You have a hydrocarbon chain with hydroxyl units on one side, and the fatty acid here, which is a long hydrocarbon chain with a carboxylic acid. And the hydroxyl unit off the carboxylic acid and the hydrogen off one of the hydroxyl units on the glycerol will actually form a water molecule, so it's a dehydration reaction, and that'll allow the oxygen and the carbon off the carboxyl unit to go ahead and form what's now called an ester linkage. And since there's 3 hydroxyl units on the glycerol molecule, you can actually pull up 3 chains. And so because it has 3, it's called a triacylglycerol or triglyceride, as they're commonly known. And you're probably familiar with triglycerides from looking at cardiovascular disease or as a cardiovascular risk factor, or something that you see on food labels. So fats separate from water because water molecules hydrogen bond to each other and exclude the fats. In a fat, 3 fatty acids are joined to glycerol by an ester linkage, creating a triacylglycerol or triglyceride. The fatty acids in a fat can be all the same or of 2 or 3 different kinds. Fatty acids vary in length, which means they can have different numbers of carbons, and in the number and locations of where the double bonds are located. Again, those 2 criteria. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds, whereas unsaturated fatty acids have 1 or more double bonds. So if you look here on the left, saturated fats, if you remember your states of matter, solids are packed more closely together than liquids. So if you look at a saturated fat, you can see that all of the fatty acid chains off the glycerol molecule here are nice straight chains. They're all fully saturated, meaning they have hydrogens every place that they can have a hydrogen bonded, then that there are no double or triple bonds, particularly no double bonds. And therefore, all the fatty acid tails, fatty acid chains here are nice and straight, and they can get compacted nice and tight together, and therefore saturated fats tend to be solid at room temperature because they can get packed so tightly together. On the other hand, unsaturated fats or oils tend to be liquid at room temperature. Why? They're unsaturated, meaning that they don't have a hydrogen every potential place that they could, that there are regions that they have double bonds, and that those double bonds actually prevent them from being fully saturated, of course. And because double bonds are shorter than single bonds, it causes a kink in the leg. So here we're seeing this would normally be a straight chain, but because there's a double bond here, it pulls it in a little bit tighter, and it causes it to kink out. Because you've got this leg on the fatty acid kinked out, as it's depicting here, it creates the potential for the need for more space between molecules, and therefore, because they're spaced further apart, they remain liquid at room temperature. Fats made from saturated fatty acids are called saturated fats and are solid at room temperature. Most animal fats are saturated. Fats made from unsaturated fatty acids are called unsaturated fats or oils and are liquid at room temperature. And plant fats and fish fats are usually unsaturated. A diet that's rich in saturated fats may contribute to cardiovascular disease through plaque deposits, but hydrogenation is the process of converting unsaturated fats to saturated fats. How do you do that? You add hydrogen. Hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. These trans fats may contribute more than saturated fats to cardiovascular disease. The major function of fats is energy storage. Humans and other mammals store their long-term food reserves in adipose cells. Adipose tissue also cushions vital organs and insulates the body. And a phospholipid, which is another class, you have two fatty acids and a phosphate group, which is attached to the glycerol. And because you have a phosphate group, it's known as a phospholipid. The two fatty acid tails are hydrophobic, which means they repel water or face away from water, but the phosphate group and its attachments form a hydrophilic head. So here you've got your phosphate group in the hydrophilic head. It's got a charge, so it's going to be attracted to water, whereas the tails are hydrophobic because they're all saturated with hydrogens, and therefore they they're nonpolar. So here you can also see it depicted. We always draw these things when we're depicting them as like a hydrophobic head and two hydrophilic tails. And then these things will self-assemble into membranes when you throw them in water because they have a hydrophilic head and a hydrophobic tail. When phospholipids are added to water, they self-assemble into double-layered sheets called bilayers. At the surface of a cell, phospholipids are also arranged in a bilayer with the hydrophobic tails pointing toward the interior. The phospholipid bilayer forms a boundary between the cell and its external environment. The final class here in lipids is steroids. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. Cholesterol, which is a type of steroid, is a component in the animal cell membranes and a precursor from which other steroids are synthesized. A high level of cholesterol in the blood may contribute to cardiovascular disease. And so here you go. This is what a steroid looks like. You've got the fused rings, lots of methyl groups, the CH3, and then a hydroxyl unit, the OH. Okay, protein functions. As you know, you drew these out the other day, so I'll go through these quickly because you've taken a look at these already for your assignment that was due. So proteins account for more than 50% of the dry mass of most cells. Some proteins speed up chemical reactions. Other protein functions include defense, storage, transport, cellular communication, movement, and structural support. So here you can see of the 8 classes, there's 4 depicted. Enzymatic proteins, their function is to selectively accelerate chemical reactions. Defensive proteins, their function is to protect against disease. Storage proteins, the function is storage of amino acids. And transport proteins, the function is transport of substances. Now that's the first 4. Okay, the next 4 for the total of 8 protein functions are hormonal proteins, which coordinate in organisms' activities, receptor proteins, which respond, creates the response of a cell to chemical stimuli, contractile and motor proteins, whose function is movement, and structural protein, whose function is support. And that rounds out the last 4 to make 8. Enzymes are proteins that act as catalysts to speed up chemical reactions. Now, enzymes can perform their functions repeatedly, functioning as workhorses that carry out the processes of life. And proteins are all constructed from the same set of 20 amino acids. As you remember, an amino acid has a amine group on one side, a carboxylic acid group on the other, bounded to a central carbon that has a hydrogen bound to it, and then your R group or your functional group. Polypeptides are unbranched polymers built from these amino acids, and a protein is a biologically functional molecule that consists of one or more polypeptides. Now, the amino acids are organic molecules with an amine and carboxyl group, as I previously mentioned. Amino acids differ in their properties due to the differing side chains, which are known as the R groups or functional groups. So again, you have the amine group on the left side, you have the carboxyl group on the right side, and you have your side chain or R group off the central carbon. It's the R group that differs. As you can see here, everything in the purple boxes is identical, but the yellow boxes, the green boxes, the pink boxes, and the blue boxes are different. Those are the R groups or the functional groups. Amino acids are linked by covalent bonds called peptide bonds. So a polypeptide is a polymer of amino acids. Polypeptides range in length from a few to more than 1,000 monomers, and each polypeptide has a unique linear sequence of amino acids. It has a carboxyl end, which is where the carboxyl unit is, and the carbon, and that's called your C terminus, and the amine end, or the amino end, is where the amine group is, because amine is predominantly nitrogen and hydrogen. It's called the N terminus for N nitrogen. Um, so here you can see you've got a peptide bond that's going to be formed here. Okay, you've got another one that's going to be formed as it gets added on. So this hydroxyl unit, this hydrogen will get dehydrated, allowing this carbon to bond to this nitrogen here. And now that's called the peptide bond when you put these two amino acids together. This is another peptide bond that was previously formed. Now over here you've got the NH group. So this is the N terminus because you have your NH2 there. And then this is your carboxylic acid side here. So this is your C terminus. Now, the protein structure and function. The specific activities of proteins result from their intricate three-dimensional architecture. A functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. So here you can see different depictions of how a molecule may be folded. We're going to talk about folding here a little bit more in detail in a second. The sequence of amino acids determines the protein's three-dimensional structure. So a protein structure determines how it works. The function of a protein usually depends on its ability to recognize and to bind to some other molecule. So here you can see you have an antibody protein on the left side. You have a protein from a flu virus on the right. You can see how the antibody is uniquely designed and shaped to fit into the bonding site on the protein from the flu virus protein. So the primary structure of a protein is a unique sequence of amino acids. The secondary structure, found in most proteins, consists of coils and folds in the polypeptide chain. The third level is the tertiary structure, and that's determined by interactions amongst various side groups, or the R groups. And then fourth is the quaternary structure, which results when a protein consists of multiple polypeptide chains. So here you go, you're looking at primary structure. You can see after it comes out of the ribosome, your polypeptide has a unique sequence of amino acids, and it's that unique sequence that gives it the primary structure. Again, you can see the amino end and the carboxylic acid end, or the N terminus and C terminus. Okay, the secondary structure is where you start getting these unique shapes and patterns called beta-pleated sheets and alpha helices. If it's coiled, it's an alpha helice. If it's a flat sheet that's horizontally and vertically aligned, they tend to form these pleated sheets like an accordion. And that's your secondary structure. Your tertiary structure is when all the alpha helices, beta-pleated sheets, and the rest of the polypeptide chain starts to fold about itself. That forms the tertiary structure and starts getting a three-dimensional shape. And then finally, if you have more than two of the tertiary structures, in other words, two separate polypeptides wrapped around each other, that forms a quaternary structure. Okay, here are some examples of the folding instances that can occur. It shows some of the different bonds you can have, like between the hydroxyl unit off of this right here and the oxygen here off of this. You can actually form a weak hydrogen bond, so that can cause those two parts of the polypeptide strand to actually be attracted to each other and fold inward. You could get sections like this where they're hydrophobic because they're nonpolar, and they repel each other and push each other away and hold it open. You can also get these disulfide bridges where the sulfurs are attracted to each other. You can also get ionic bonds between positive and negative charges. So different levels of strength, different points of folding in the structures. This is a quaternary structure here. You can actually see that there's multiple colors here. There's purple and light blue. This is actually hemoglobin, the molecule that carries oxygen in your blood. The heme groups have an iron atom inside of them. There's 4 polypeptides here. There's 2 alpha and 2 beta subunits, and this is a quaternary structure because there's more than one polypeptide chain folded together here, and they're trying to depict that here with the 4 different pieces, but 2 separate colors. The primary structure of a protein is its sequence of amino acids. The primary structure is like the order of letters in a long word. Primary structure is determined by inherited genetic information. Now, the coils and folds of secondary structure result from hydrogen bonds between repeating constituents of the polypeptide backbone. Typical secondary structures are a coil called an alpha helix and a folded structure called the beta-pleated sheet. Tertiary structure, the overall shape of a polypeptide, results from interactions between R groups rather than interactions between backbone constituents. And these interactions include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waal interactions. Strong covalent bonds called disulfide bridges may reinforce the protein structure. Quaternary structure results when two or more polypeptide chains form one macromolecule. Collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. Hemoglobin is a globular protein consisting of 4 polypeptides, 2 alpha and 2 beta subunits. A slight change in primary structure can affect a protein structure and ability to function. Sickle cell disease, an inherited blood disorder, results from a single amino acid substitution in the protein hemoglobin. The abnormal hemoglobin molecules cause the red blood cells to aggregate into chains and to deform into a sickle cell shape. This is depicting it here. You can see the substitution in the sickle cell there at number 6, valine, where it should have been glucine or glutamine. And then as you look across, you can see what the change in shape means, and it changes the structure of the protein, and therefore it changes the red blood cell shape. In addition to primary structure, physical and chemical conditions can affect structure. Alterations in pH, salt, concentration, temperature, or other environmental factors can cause a protein to unravel. This is called denaturation. A denatured protein is biologically inactive. So there's a picture depicting denaturation and renaturation. It's hard to predict a protein structure from its primary structure. Most proteins probably go through several stages on their way to a stable structure. Diseases such as Alzheimer's, Parkinson's, and mad cow disease are associated with misfolded proteins. Scientists use X-ray crystallography to determine a protein structure. Another method is nuclear magnetic resonance, NMR spectroscopy, which does not require protein crystallization. Bioinformatics is another approach to prediction of protein structure from amino acid sequences. I think this is the last section. Nucleic acids store, transmit, and help express hereditary information. The amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. The genes consist of DNA, nucleic acid made of monomers called nucleotides. And there are 2 types of nucleic acids. There's deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA provides direction for its own replication. It directs synthesis of messenger RNA and, through messenger RNA, controls protein synthesis. This process is called gene expression. So here it's showing the messenger RNA strand in the nucleus as the messenger RNA travels through the nuclear pore into the cytoplasm, and then as it travels through the cytoplasm to a ribosome, either on the endoplasmic reticulum or a free-floating ribosome, in order to be read and translated into a polypeptide. Each gene along a DNA molecule directs synthesis of a messenger RNA molecule. The messenger RNA molecule interacts with the cell's protein synthesizing machinery to direct production of a polypeptide. The flow of genetic information can be summarized as DNA makes RNA and RNA makes protein. Nucleic acids are polymers called polynucleotides, and each polynucleotide is made of monomers called nucleotides. Each nucleotide consists of nitrogenous base, a pentose sugar, and 1 or more phosphate groups. The portion of a nucleotide without the phosphate group is called a nucleoside. So the nucleoside is the nitrogenous base plus the sugar. There are 2 families of nitrogenous bases. The pyrimides, which is cytosine, thymine, and uracil. They have a single 6-membered ring. And then there's the purines, adenine and guanine. They have a 6-membered ring fused to a 5-membered ring. So in DNA, the sugar is deoxyribose, and RNA, the sugar is ribose. So the nucleotide is equal to a nucleoside and a phosphate group. Okay, nucleotides are linked together by a phosphodiester linkage to build a polynucleotide. A phosphodiester linkage consists of a phosphate group that links the sugars of 2 nucleotides. These links create a backbone of sugar phosphate units with nitrogenous bases as appendages, and the sequence of bases along a DNA or messenger RNA polymer is unique for each individual gene. DNA molecules have 2 polynucleotides spiraling around an imaginary axis, which creates the double helix. And the backbones run in opposite directions from the 5' end to the 3' end. This is called anti-parallel. One DNA molecule includes many genes. Only certain bases in DNA pair up and form hydrogen bonds. A always will bond with T and G will always bond with C. This is called complementary base pairing. This feature of DNA structure makes it possible to generate two identical copies of each DNA molecule in a cell preparing to divide. So RNA, in contrast to DNA, is single-stranded. Complementary pairing can also occur between two RNA molecules or between parts of the same molecule. In RNA, thymine is replaced by uracil, so A and U pair. While DNA always exists as a double helix, RNA molecules are more variable in form. Once the structure of DNA and its relationship to amino acid sequence was understood, biologists sought to decode genes by learning their base sequences. The first chemical techniques for DNA sequencing were developed in the 1970s and refined over the next 20 years. It's enlightening to sequence the full complement of DNA in an organism's genome. The rapid development of faster and less expensive methods of sequencing was a side effect of the Human Genome Project. Many genomes have been sequenced, generating large sets of data. Bioinformatics uses computer software and other computational tools to deal with the data resulting from sequencing many genomes. Analyzing large sets of genes or even comparing whole genomes of different species is called genomics. A similar analysis of large sets of proteins, including their sequences, is called proteomics. Sequences of genes and their protein products document the hereditary background of an organism. Linear sequences of DNA molecules are passed from parents to offspring. We can extend the concept of molecular genealogy to relationships between species. Molecular biology has added a new measure to the toolkit of evolutionary biology. And that should do it for Chapter 5. There's some great diagrams at the end there to refer to. And have a good day.